Bacteria, primitive yet surprisingly complex and resilient. Their seemingly limitless capacity for mutation and adaptation constitutes the major challenge to the research effort to control and to cure bacterial infections. To better understand this challenge, let's examine the most familiar of all pathogenic bacteria, Escherichia coli. The outer surface of a gram-negative bacterium, such as E. coli, is a tough barrier, which maintains the integrity of the organism and aids its survival in hostile environments. An amorphous, slimy polysaccharide layer encapsulates the bacterium. This capsule, secreted by the bacterium, facilitates colonization and protects the organism from phagocytosis. It may also prevent the entry of certain antibiotics. Long helical projections called flagella propel the bacterium. The fine hair-like projections emanating from the surface of the cell are the pili, which enable the bacterium to adhere to surfaces in its natural environment and in certain disease states. A cross-section of an E. coli cell reveals two membranes separated by an area called the periplasmic space. Unlike most biological membranes, the outer membrane of E. coli is an asymmetrically organized lipid bilayer. Lipopolysaccharide forms most of the outer leaflet of this membrane. This LPS determines the O antigen character of the cell and is required for pathogenicity. Its lipid portion, lipid A or endotoxin, is highly toxic to mammals. The polarity of the lipopolysaccharide is thought to prevent many antibiotics from entering the cell. Other surface components are phospholipids and some 20 to 30 types of proteins that play a role in determining membrane permeability. The most abundant of these are porins, which form channels for rapid transport of small hydrophilic nutrients. Certain antibiotics also pass through the porin channels. Just inside the outer membrane lies the peptidoglycan, the rigid structural framework of the bacterium. It is a single giant molecule consisting of glycan chains cross-linked by peptide bridges. It is covalently attached to lipoprotein molecules embedded in the outer membrane. The periplasmic space contains a host of specialized proteins, including binding proteins involved in chemotaxis and the transport of nutrients. There are also various hydrolytic enzymes, including beta-lactamases, which are largely responsible for resistance to penicillins and cephalosporins. Many different types of beta-lactamases, both chromosomal and plasmid-mediated, have been identified in gram-negative bacteria. The enzyme inactivates the antibiotic by hydrolyzing the beta-lactam ring. Below the periplasmic space lies the inner or cytoplasmic membrane, a typical lipid bilayer with a large protein content. On its outer surface are the enzymes responsible for the synthesis of peptidoglycan. Certain of the enzymes are apparently the lethal targets of beta-lactam antibiotics. Because they bind covalently to penicillin, they are commonly referred to as penicillin-binding proteins, or PBPs. E. coli has at least 7 PBPs. PBPs 1A and 1B are responsible for the bulk of peptidoglycan synthesis. Either of them can catalyze both the formation of the glycan chains and the formation of the peptide crosslinks. PBPs 2 and 3 are thought to catalyze similar reactions for special purposes. Without PBP2, the cells lose their rod shape and become spherical. Without PBP3, they are unable to septate or divide and grow as filaments. A complex interplay of these enzymes with PBPs 4, 5, and 6 is required for normal growth. The inner membrane is the last barrier to the heart of a bacterium. Most of the metabolic processes of the cell occur either on the surface of this membrane or inside it, in the cytoplasm. Complex and relatively specific transport mechanisms operate to transfer molecules across the inner membrane. Antibiotics, which act in the cytoplasm, must somehow exploit these mechanisms. The genetic directions for all cell activity are contained in a nuclear body or chromosome in the form of deoxyribonucleic acid, or DNA. DNA serves two important functions. First, it transmits genetic information to bacterial offspring during cell division. And second, it directs the synthesis of cell proteins. Genetic information is encoded in the bacterial chromosome by the specific sequential arrangement of the 4 organic bases: adenine, guanine, thymine, and cytosine, in groups of 3, or triplets. Each triplet in a structural gene ultimately specifies one amino acid in a protein. In E. coli, the DNA takes the form of a closed circle of the familiar double helix, with 2 complementary strands held together by some 4 million pairs of bases: adenine paired with thymine, and guanine with cytosine. Genetic information is transmitted to progeny cells by replication of the DNA molecule, in which new strands are synthesized in strict complementarity to the templates provided by the 2 parent strands. The unwinding of the helix, which is necessary for replication, is mediated by an enzyme called DNA gyrase, one of at least 13 proteins involved in replication. Gyrase first breaks both strands of the helix and then rejoins them on the opposite side of an overlapping portion of the DNA molecule. This results in supercoiling of the DNA molecule, which facilitates unwinding of the helix. The quinolone class of antibacterials apparently act by inhibiting DNA gyrase. Gene expression begins with the transcription of a DNA sequence into a strand of complementary messenger RNA by RNA polymerase with the help of other proteins. As the strand of messenger RNA separates from DNA, its message, a series of triplets of bases or codons, is translated into the amino acid sequence of a protein by complex organelles known as ribosomes. The amino acids must be activated for protein synthesis by linkage to transfer RNA molecules. The ribosome matches an anticodon in the transfer RNA to the codon in the messenger, and then transfers the growing end of the polypeptide chain to the amino group of the new amino acid. The ribosome then moves on to the next codon and repeats the process, eventually forming a protein with the sequence specified by the genetic code in the DNA. Many antibiotics, such as chloramphenicol, tetracyclines, and aminoglycosides, interfere with microbial protein synthesis. The aminoglycosides seem to cause a mismatch of the bases in the codon and anticodon. Synthesized proteins serve a multitude of purposes within the bacterial cell. They include, for example, the enzymes that catalyze the myriad vital metabolic processes. One such protein is dihydrofolate reductase, an enzyme that is essential for the formation of tetrahydrofolic acid, which is required for the synthesis of thymine, and therefore of DNA. The antibacterial agent trimethoprim acts by inhibiting dihydrofolate reductase. Sulfonamides block another enzyme in the same metabolic pathway. When both trimethoprim and a sulfonamide are present, synergy may result. The challenge of antibiotic research is to better understand the bacterial cell, its physiology and functions, and to exploit this knowledge in order to find new ways to destroy pathogenic bacteria. The answers to many of our infectious disease problems and the clear understanding of cell structure, mechanisms of resistance, and modes of action of antibacterials. We hope you've enjoyed this presentation, a stereoscopic study, E. coli, brought to you by Roche. Please consult with the Roche representatives for further information. Thank you.
Need to get going?
Okay.
Okay. You ready? Here. We're going to talk here for just a minute. Koko, I have a question. Yeah, I have a question. You know Mike is the devil. I didn't ask you about Mike. What happened to your kitty ball? Trouble? What happened to Ball? Guys, I'm sorry if Koko love. An attention visit. He doesn't visit you anymore. What happened? Do you know? Do you know what happened to Ball, the kitty? What happened? Me? Gorilla? Good. You are good, honey. You are good. You're a good gorilla. Yes, you are. I'll tell you what. When you get another kitty, what kind would you like? You tell me what kind of kitty you would like if you get another one. That? That kind. Okay, she's pointing to this one. You'd like that kind. All right. We'll get you another kitty like that. Is that okay? That's nice. Okay. You sweetheart. All right. Do you want to do any more? Should I ask her how she feels about kitties? Okay, I have one more question, then we'll get that. One more thing. How do you feel about kitties? Cat gorilla have visit. Koko love. Good. She'd like to have another cat visit. Do visit. Do. Okay. We will. In a little while. Trouble. Trouble might visit. What? Oh, honey. Oh, you're so eager for that. We're going to bring you a new kitty. Okay. All right. Not anymore? Okay. We were going to ask you about these kitties. This picture. Was there reflection? Tell me about that kitty. Okay, how about this one? And we can talk. We can talk about cats and how your feelings are about cats and how you feel when you see kitties like this. Yes! You know what she just said? This one looks like Stink. And it does. It looks just like Stink. It's the same color. Look. We had a cat named Stink that looked like that. He was brown. Bright. And this one, who does that look like? Need that. Oh, you need that red. We talked about a red kitty. Yes. Lips Visit cat there. Yes. Does this kitty... Koko, turn around a minute. How's your face doing? Doing good. Who's this kitty? Me need that. Oh, honey, we'll try to get you a kitty like that. Oh. This is the one that I was interested in. This one or this one. Do you remember about kitty like this? Do you remember? Kitty visit that gorilla Koko love. Yes, a red one. I hope. Trouble, attention, honey. We have been having trouble getting you a new kitty. We've been trying very hard, and it didn't come when it was supposed to, but we're trying more, and we'll get you one soon. We'll get you one in a little while. Do you have any thoughts about this one, though? That one's red. One in red like that. Well, I think maybe that we can fill your order. We can help you. So, how do you feel? Really have nipples. You have nipples. Good. Maybe for the kitty, baby. Okay. All right. I think we're ready for our next. Here, let's see here. Here, Koko, wait. Would you sit here for just 1 minute? I wanted to tell you, how did you feel when you lost Ball? How did you feel when you lost him? Trouble visit. When he died. Remember when Ball died? How did you feel? Sorry. Koko, love. Good. Were you sad? What? Frown lips. Bad. Frown. Oh, sad. You were sorry. Okay. What have we got here? What? Nice? Nice. Sorry. Gorilla. We have a nice sorry gorilla. What's in the box? An animal sleeping? Okay. There. A new kitty. Something in your eye. Get it out? Koko, let's not worry about it. Damn. Hard to carry a baby, isn't it, when he doesn't sing? We have a little dust on you here. Maybe you could have the kitty talk on that. You're gonna talk. Kitty talk. Well, Kitty will learn how to do that. I'll move this back where it was, okay? Give him a kiss. Oh, no. Show him your dinosaur? Mm-hmm. Let's show him your dinosaur. Boy, I bet that'll scare him. Oh, show him how it bites. Love that baby. Are you being gentle? Gentle, Koko. He's tickling your stomach, isn't he?
Koko, what? I think you're being... He doesn't like to be up in the air, I think. To give him support.
How about you thought of a good name? What's a good name for that kitty? What's a good name? Yes, his eyes cry. He has a little eye problem. We're fixing it though. Do you like your kitty? You're happy. What was that? I want you to be gentle, Koko. Koko, this is the kind of thing he doesn't like so much. Doesn't like so much to be held up. That's better. That's better. Being gentle with his little paws. That's it. Yeah. Why is he obnoxious? Obnoxious in that case. What? Why? Because he cries, maybe. Candy. You want me to get you some? Okay. First you name him, and then I'll give you something special. What's a name for him? A name. You think about it. You're not going to think about it? We've all been thinking of a good name. Got a nice tummy. Put him away. All right. And what about the new cat? Let's think of his name. Trouble, surprise, visit. Oh, honey. Is it a trouble surprise visit? Oh, I don't know. Can you think of a good name? She doesn't know. Okay, well, if you don't know, I suppose that's all right, too. You can think a little bit longer for a name for him. You know what Mike called him? Yeah, actually this is green. Maybe red. No, I don't like that. Mike called him. You know what Mike called him? Mike thought banana was a good name.
Banana's nice. Banana. Okay, how about a better name? Can you think of a better name than banana? Banana. Better name. Bad Koko. Honey, you're not bad. You're good. No, you're not bad. Bad. Oh, it's bad because she doesn't know the name. Okay, banana, and she said bread. Well, okay, we can take our time thinking of a name. That was Mikey's. Oh, these are some of the letters from the children. Let's see if we can find a picture. Oh, I thought we'd given these letters back. These are such nice letters. Koko, look!
Do you want to hold them up and I can give them a place? That's good.
Look at these nice letters. That's a gorilla. They wrote in red. Yes.